So hello, welcome to this event by Cancer Patient Lab. I'd like to introduce our speaker today, who is Dr. Joe Lennerz, who is the CSO at Boston Gene. He's going to talk to us today about integrated diagnostics and then also lead a discussion on the topic. Now, he has experience as medical director. Please, everybody, mute your phones. He has experience as medical director at Massachusetts General Hospital, Harvard Medical School, prior to becoming CSO at Boston Gene. And at Boston Gene, he works on combining multimodal data to guide cancer treatment. So thank you for being here, Dr. Lennerz. And as noted, we typically start off with the presentation. I don't know if you have slides. If you do, I'll allow you to share the screen. And if not, we'll just let you talk.
Perfect. Thank you, Roger. If you could give me permission, then I prepared a few slides and I want to quickly go through that to just sort of set the theme. So as we just in this brief chat before the recording discussed, integrating diverse test results is really one of the key challenges or gaps. And to address this today, I tried to separate this into four things. And I want to start with what do we actually need to navigate this very complex landscape of tests. Now, as you know, precision oncology is not only a buzzword, but it is a plethora of solutions. For example, just in the space of cell-free or circulating tumor DNA, RNA, or biomarkers, you see here there's multiple different use cases just of that one technology. And providers as well as patients are faced with this, diverse set of solutions. When it comes to down practically, when looking at, let's call it a certain challenge, it is that on the one hand, the guidelines and approvals are constantly changing. I show you here a part of a non-small cell lung cancer NCCN guideline page. There's constantly new findings coming out in the literature. And then, of course, the daily practice sometimes looks like this, where, individuals have to manage and navigate elements. So how do we address this? So, of course, as Boston Gene, we have many different solutions. And I want to show you that, when looking at this, there's different almost like cards in a Rolodex that you can pull out when you want a certain lab technology to be applied to something. To make it a little more concrete, I want to show you our, let's call it ecosystem of tests that we have in a sort of interactive fashion, which is basically there are many different elements. For example, in next-gen sequencing, we perform whole exome sequencing at the DNA level, at the RNA level, and then, of course, whole genome sequencing, for example, or single-cell RNA-seq. And that forms one column of services. But as just outlined, there's liquid biopsy. We're very, very specialized on immune system profiling and also on tissue imaging. And in each of these, there's a raw data layer, there's a processed data layer, and then, of course, advanced analytics. And I come to that in a second. So for the discussion, I thought that navigating this landscape is that every single provider has to build their own portfolio of solutions. They're sometimes built with a purposeful redundancy. For example, to be able to know, is a marker really expressed in the cell that we want? Can we verify this at the DNA level, whether it's a somatic mutation or germline? So what I'm trying to say here is orthogonality is key, and decision-making is often relying on more than one data point. So in a way, when we transverse, let's say, this gap from real-world data to real-world evidence, this landscape becomes a resource. And if you're resourceful, you can actually turn this, let's say, additional precision oncology or precision diagnostics really into action. So this is not just learning something to have CME points, but to actually, become, let's say, a better physician, better oncologist, and have actionable results. So the second part that I wanted to share is, how do we actually integrate findings? And one thing that I thought of portraying to you is sort of a shapeshift in this landscape that we're currently witnessing. So what's shown in this very simple graph as a key message is on the bottom, you see there's the system integration. And what I mean with that is the workflow established, which would be closer to here, or do you need more administrative and IT resources that may not be as integrated as we wish? So that would go further to this side. And then on the y-axis is just data complexity. Now, traditional diagnostics would rank somewhat here. Traditional diagnostics is established, so system integration is there, but the data complexity is not as high as we might want to have it. If you combine traditional diagnostics, let's say radiology, surgical pathology, traditional histomorphology, immunohistochemistry, and maybe some molecular testing, you might be in this intermodality range where, depending on the care pathway, you combine certain things together. If you combine them with intent and align it at the system integration as well as the data complexity, some people call that integrated diagnostics. But what the field is really trying to do now is to move past that and really talk about multi-omics. I can show you what that looks like, at least for Boston Gene, and I wanted to show you two examples. One is that when we test clinical patients, we usually do a triple exome, which is DNA, RNA, and a germline. We take into account the treatment timelines and then current recommendations. So you can say there's a patient section, there's the context, the actual context of care in that particular patient. I show you that in a second. Then the actual findings, and then selected treatment options and even a location-specific trial match. That level of integration into one-page report may not be sufficient to some really driven or research or cutting-edge oncologists. So we have a lot more pages, but we try to make it as concise as possible. And we call the thing a portrait rather than a profile. And I'll show you an image about that. So as an example, I want to show you that these abstract notes, clinical notes text, that many oncologists spend a lot of time making this as concise as possible. We extract using a unified medical language abstraction and extract the concepts into these treatment timelines. And these treatment timelines have one underlying principle, that is that simplicity is key. So you see here, this is not a timeline exactly, but the key events get an equal distant element so that you can get a very quick overview. And this is, for example, used in tumor boards, etc. The other part that I want to point out is that we customize our reports to the setting. As an example, on the left is that breast cancer report that I just showed you, and on the right is a lymphoma report, where jointly with some of our lymphoma experts, we restructured basically the layout to make that really personalized, not to the provider, but to the setting. So if you see a Boston Gene report and it looks completely different from cancer to cancer, that is caused by an optimization for that specific setting. So in other words, what we portray is that we switch from a profile or tumor profile to really a portrait to really capture a bit more of what the cancer is about. Now, one of the things that we do is, of course, we use advanced analytics. And I want to demo this to you real quick. So when we collaborate with our drug developer partners, pharma partners, or researchers, we offer a comprehensive set of solutions where you may have data, let's say, for example, data like DNA-seq, RNA-seq data, or imaging data. These colors represent the frames in these figures. And then when looking over this large board, what you can see is that for certain elements, let's say someone wants to correlate protein expression status with transcripts or cell segmentation or classification of neighborhood, nearest next or nearest neighbor analysis, you can click on the bottom left and that will lead you to a peer-reviewed publication, including our team members that have access to the GitHub code so that we can reconstruct these things very quickly. So the second part, when it comes to integration of findings, is, we move from profiling to portraits, more comprehensive multimodality solutions, and we take the treatment context into account, and I showed you the advanced analytics. Now, the next element is what is the right test at the right time? That is, of course, a very complex and challenging question, and I want to introduce this through a paradigm shift that happened over the last decade. From an initial identification of EGFR, where multiple targeted therapies emerged from one single biomarker, to a companion diagnostics approach that, Foundation Medicine in 2017 has put forth, where a multitude of markers linked to a multitude of therapies, all the way to 2020, when an entirely different approach for precision diagnostics has emerged, where multiple biomarkers converge on immune checkpoint inhibitor therapy. You've seen that basically the change in landscape of companion diagnostics has now resulted in something, when I largely oversimplify this, that some cancer need very comprehensive genotyping, while other cancers primarily need, of course, just a certain set of markers or combinations. And you may say, well, Joe, this is wrong because you didn't even put the stage here and you didn't even put the specific setting here. And if the patient had targeted therapy, it would look entirely different. What I'm trying to say is that many groups have started implementing what is called order sets. Here's an approach that was published by Dora Diaz-Santagata in 2022, where an interdisciplinary team got together and decided for certain tumors, certain test combinations are needed. Some people call that order sets. And I wanted to give you a quick analogy, and the right diagnostic at the right time, you could call it R2D2, and it might be actually really complicated to find a message in that. What we realized is when we do triple exome, we have the ability to add all kinds of signatures, all kinds of outputs for the different tumor types. So what this has resulted in is a cancer-specific combination of elements. So a PAM50 signature in breast cancer, you could produce bioinformatically to a prostate cancer or kidney cancer, but that wouldn't make any sense to classify based on these signatures. So what this illustrates is basically a flickering map of new signatures and new updates that would come out relatively quickly. So homologous recombination deficiency or DPYD testing is currently recommended in some, but not all tumors. So I want to illustrate this, that we've moved really to a personalized diagnostic stage in precision oncology. Now, that sounds promising, but what the reality is, and I want to just mention this, is from order to a procedure and payment, there's the thing called prior authorization, and in clinical reality, it looks more like this. So why I want to emphasize this with this group is that, you're all aware that there's a financial sustainability problem, and I just wanted to point this out, that the right test at the right time, of course, is what I just termed personalized diagnostics, and sometimes not doing testing can be actually meaningful. And then this weird letter cryptic code here means that prior auth, denial, appeal, testing, denial, appeal, letter of medical necessity, and medical exemption status claims is probably something that, put here that we need to uphold standards and order sets and IT integration is that. I want to end with something about finding the right partners and collaboration. And I want to show you something that is fairly unique, and in this group, it might actually hopefully resonate. A few years ago, the laboratory around John Haymack at MD Anderson published a small cell lung cancer classifier that distinguished 4 prognostic groups, and it had a huge promise that some groups actually, based on their biochemical and biology, might respond different to different therapies. We collaborated, and the plan was to roll this out as a classifier, meaning we converted this into a clinical test. And then only, in this case, 2 years later, we have a CLIA and CAP approved test that is rolled out clinically and now serves as a phase 2 clinical trial enrollment stratifier in a Southwestern Oncology Group trial. Now, this level of collaboration where certain data sets and literature is used to build a tool that is through public cohorts as well as a tuning set that goes really into the clinic and helps improve standard of care is somewhat very unique. So I want to mention as sort of a final statement here, this find and collaborate is things like this meeting here, where different groups, different stakeholders come together and really collaborate. And one quick thing for those on the call who haven't met their pathologist, please make a call today, call your pathologist and say, hey, I just want to introduce myself if you haven't done so. I know many of you have. And I want to end, of course, by thanking you and then saying that these 4 points hopefully will form the basis for an interesting discussion today where, navigating the landscape, the integration of findings, finding the right tests at the right time, and then, of course, finding and collaborating will hopefully help integrating diverse test results for cancer care guidance. If you have questions, please feel free to reach out. And with that, I hand it back to you, Roger. Thank you very much.
Okay, thank you. Yeah, if you have questions, why don't you go ahead and use the raise hand feature. I want to start off with something because I actually am a customer. I used your services. And the challenge that we had, you ended on kind of the biggest issue for me was just getting tissue from the pathologist. And that was 3 years ago. Have things improved in that area? Is it have the major institutions now familiar with testing and companies like Boston Gene? And because I had to go to, with the help of Rihanna, we had to go to just extraordinary lengths, and I mean extraordinary lengths, to get my tissue. You don't even want to know. I'm just wondering, what's it like now?
So absolutely important question. Tissue is the issue, and getting the right tissue at the right time, I didn't put this there, is definitely, it continues to be a problem, but the situation has improved. I give you 2 or 3 reasons why it continues to be an issue. The value of molecular testing is continued to be recognized. And a common setting is, for example, with patients with metastatic disease to the liver, where the oncology team knows 100% this is breast cancer. It looks like it. We've seen it. Even at the same institution, it might be that the pathologist feels compelled to work this case up, cut through the sections, that gets additional immunohistochemistry, and some people call that wastes tissue in an attempt to reconfirm the diagnosis that it was really breast cancer. There was never a question that this was not breast cancer, but then very frequently we end up not having enough tissue for molecular genotyping when the whole purpose of the biopsy was that. Now, if then the molecular testing company is external, most of the commercial vendors have tissue procurement teams that I think, Roger, you described, where you had to have someone make calls, ask for the right block. Then you may get a sample that doesn't contain tumor, or you may get blank sections, et cetera, et cetera. So I'm just describing this in detail because it is not, or it was not that clear to everyone why that is. Practically, if the oncologist indicates that specifically that the biopsy is taken for identification of new therapies, that is often working. So we witnessed that helps tremendously if the clinicians is helping with that. The second part is many samples do not have enough tumor content. Now in pancreatic cancer, it depends totally on the size of the sample. The biopsy might be a fine needle aspiration or a smaller core needle. And not every tumor consists just of tumor cells. And that is often also a problem, that even despite finding the tissue finally, that there's not enough tumor material on the slide to actually extract enough nucleic acids to do the testing. So those are, some of the elements. How to overcome that is by building really strong relationships with the laboratories and the oncologists, and of course, sharing with the team also what the benefits are of the results. That positive reinforcement definitely works, and doesn't cost much to send a team, hey, thank you for sending the blog. This was really helpful. Or thank you, we identified an NTRK fusion. The patient now received therapy. Those tiny little, signals of gratitude actually lead like a long way. I see a question from Emma about, the issue about
Kind of leaning into what you were just talking about, but specifically, what if you do have a tumor with low content? Like pancreatic is often less than 20% actual tumor tissue, and the rest of it is fibrous. Can you analyze that?
Yeah, so we worked on a solution to go down to 1 to 2%. The main problem is that, going live clinically with something like that provides a little bit of a liability, and the validation is actually a problem. So while analytically we can see variance at that low frequency, the question is how can we reliably validate this? So you can come up with a certain validation approach, but currently we do not offer that clinically live yet, but we're working on a solution that would go down to 1%. Now, is that always analytically valid, and can we always claim that it's clinically valid? Probably not. So we probably need to add a disclaimer. But for tumors like, pancreas, where a common driver is always there, that like a lead indication that we do have tumor is very helpful. Gets a little bit in the same realm as liquid biopsy testing, where if you do not see a variant, is it truly not there? Meaning, is it a pertinent negative, or did we just not detect it? And that really continues to be a challenge. And we have some solutions to overcome that, but it's actually immune profiling, but definitely a challenge. Great question, Emma.
Okay, I see another question in the chat. How do you ensure the reproducibility of your results?
So I hinted at that a little bit. So with multi-omics, one of the key elements is that you need certain anchor points in one, let's call it modality, that can be replicated in another modality. I'll give you an example. Let's stay with pancreatic cancer. Let's say you have a KRAS G12C mutation that you identified at the DNA level. And now you want to do expression profiling, let's say using RNA sequencing data. One sanity check is, of course, to verify that that same KRAS G12C variant is present at the RNA-seq data set. Another very common integration for reproducibility and, let's call it data integrity is it the same HLA genotype between, let's say, germline testing and somatic? So these anchor points between the different data sets that serve as a linker to ensure that, yes, these findings are reliable are actually absolute key. So some people call that just quality metrics in multi-omics, but if you do not have those, it gets very dicey. So especially when you do gene expression profiling and you have copy number assessments, that's something to just verify and be aware of.
Okay. Another question, is RNA testing actionable with regard to prostate cancer? Are any therapies FDA approved for RNA results?
So that's a very, very good question. So there are currently, to my knowledge, no FDA-approved therapies. However, there are certain findings from RNA-seq and prostate cancer that are really important. One is 10 plus ERK. So finding that fusion, you can use, of course, DNA sequencing and find a breakpoint somewhere, but the other part is to get the actual 10 plus ERK fusion transcript that you can identify with RNA-seq, and then you at least know that that is that cancer. It's very, very common in prostate cancer. There's a few individual case reports in bladder cancer. I highly doubt that. I haven't actually seen that convincing, but that might be exceptionally rare in the same genitourinary region. Why that is actually important is if you find a few years later, because prostatic cancer is such a long and protracted disease, you find a lesion, let's say in the retroperitoneum or in the spine, you can reassess whether that is truly harboring still that same fusion, so you can establish sort of a phylogenetic relationship. The second finding that's important or of interest, and a few years ago, there was a paper in the New England Journal of Medicine by Emmanuel Antonarakis, I think from Johns Hopkins, where a certain alternative splice form of the androgen receptor called ARV7 was described in the context of castration resistance. Now, this is very interesting that the androgen receptor, composed of different exons, can, under this drive of castration resistance, splice to a cryptic exon. I think it's cryptic exon 3, and that specific isoform is called ARV7. that element cannot be really distinguished because it's a splicing event and you need RNA-seq for that. Now, the data is not 100% clear whether ARV7 always occurs in the setting where then next generation anti-androgens work like abiraterone and enzalutamide, but these alternative splice isoforms in is one of the key applications. I know that a lot of people work on prostate cancer expression profile-based signatures. As I think this community is aware of, Gleason grading remains to be a key element for distinguishing high-grade, low-grade prostate cancer. But that is an ancient approach, certainly reliable, highly inter-observer variable. But I know many groups are working on RNA-based signatures to hopefully provide other prognostic indicators. I'm seeing a few other comments here, but yes, so I would call it emerging evidence for RNA-seq in prostate cancer as a quick summary. Antonakis has led a discussion with yeah, perfect. No, I hope I didn't butcher his name, but I remember the paper very well. ARV7 is digital there or not there or analog? Can it be predicted? Yeah, a lot of work on this. Even the question like how to detect ARV7 is questionable. There was some work on can you detect this in the blood? Can you use this for treatment monitoring, almost like a second independent element to PSA tracing? So great question, Ross, but I must say I have not seen anyone offer that all the time. So traditionally, we've done some internal work that anything over 8 reads splicing to the cryptic exon seems to be reliable in terms of, yes, it's present, but that's highly inter-assay dependent. And yes, you could quantify it, and some tumors really have extremely high ARV7 expression, while others just have it. So it's probably, in part at least, a subclonal event.
So another question, how do you correlate CNV data with biological consequences? How many extra copies of a given oncogene can you link with an oncogenic effect?
Oh, very, very, very good question. So first of all, that CNV stands for copy number variation. And copy number variations can either be gains or losses. So copy number gain or CNG or copy number loss or CNL are the two types of copy number variants. Now, the work by John Frady, published in Nature Genetics, illustrated that actually as humans, we have in our normal cells one element that contributes to our genetic variety, copy number alterations, and they can be normal. So certain genes or certain regions, we just differ human to human by having different numbers of copies of our genes. That's actually very, very important to keep in mind when describing copy number alterations, because if you only look at the tumor and you don't have the corresponding normal, you might actually overinterpret a intra-patient copy number alteration that is just present and normal as a copy number gain. That's just a start to say when you determine a cutoff, it should actually be made in the context of that patient when we talk about this. For certain copy number gains, when they're in fluorescent in situ hybridization, you can see actually fluorescent dots per gene when the gene is hybridized. In some patients and some tumor cells, it's too numerous to count. And it becomes almost like mundane to say, oh, definitely this gene is amplified. That's typically the case, for example, in EGFR amplification in glioblastoma or in HER2 amplification in breast cancer. But when it comes to determining a cutoff, to say, where's that cutoff? There's two things interfering. Number one, if a tumor has low-level gene amplification, it can actually be a high-level amplification, but the tumor just contains a lot of inflammatory cells. So it really distinguishes between, is the high-level amplification or mid-level amplification caused by the cells just having a few copies that were gained, or is it an artificial dilution by normal cells, meaning a lot of cells with no amplification? So how we work on that or how we overcome that is with two things. Number one is by having tumor normal. I mentioned earlier, we always do triple exome. So we have the reference normal, and then we know the exact, let's call it copy number in the tumor. And that is very helpful, especially to identify, let's say, heterozygous loss or loss of heterozygosity or genotype loss or gains, even low-level gains. And then we can verify this, and this gets to the point of how do we know whether these copies actually have an oncogenic effect? We find that certain genes are gained in terms of copy number, but then we do not see an expression of that gene. So since we always have the transcriptome as well, meaning the RNA level, we can check whether that particular transcript is also enriched. So that's, again, a little bit of that orthogonal verification that I mentioned earlier. But it definitely depends on which gene it is and what the context is. Now, one of the key elements, for example, in mesothelioma and in a few other cancers is the CDKN2A and MTAP loss, which becomes more and more important, especially in that locus. And so it's definitely a relevant question. And if you have multiple elements, germline, tumor, as well as RNA, it gets a little bit easier, but it's certainly a challenging topic. Sorry, long answer to a simple, seemingly simple question.
Okay. Next question is, when data from multiple modalities are used to develop a biomarker, could there be challenges to validate because of the potential requirement to data from these modalities for each patient in the study?
Oh, also a very, very good point. So this gets to the point of, is simplicity good enough? In other words, if you know what a biomarker is and you have a biomarker, let's say, as a response predictor or a secondary endpoint, some oncologist or drug developer likes to call that correlates. If you have a simple set of correlates that fulfill the purpose, why go for multimodality? That is a certain sentiment in the field. The complete opposite is if we plan a multi-million-dollar study and we get additional data, and we might actually get one additional factor that might make our prediction a little bit better, you almost have to go for it. Because, when you plan such gigantic efforts, regulatory, the planning, the preclinical data, the first in man, maybe phase 1, phase 2 transition, it becomes sort of a risk mitigation element. And, we would argue for, especially in the earlier phases, to get a maximum amount of data out of the sample. In other words, if you get a DNA panel from your sections and then later you find, maybe in this breast cancer we would need expression profiling subtyping to check whether, a certain response tracks with that, we argue that not losing that additional layer might be a missed opportunity. Now, how do you validate this? So we have some good data with some of our pharma partners that, especially when the trials start and you gain the initial insights, that these early phase, let's call it monitoring events or early events, are incredibly insightful, because at that time you want to have all data together in the specific treatment context. The other part is also, which is frequently forgotten. If a patient goes on trial, many patients progress and have to go on a subsequent therapy. And it is very important to know it's not always the first line or the second line being on trial. It is also, can this additional multimodal result potentially inform my subsequent line of therapy? And so those are some of the elements. It's almost not a financial argument anymore. It is more, is this going to be useful? And when taking those elements into account, I would definitely opt for multimodality. Now, very important, in reverse, when we talk about routine clinical practice and application of these, let's call it high complex solutions, from a payer perspective, we can't make that argument yet because this is all emerging evidence. We do not know that it's proven. So I fully understand the reluctance of some payers or from CMS to say, yeah, we can't just get everyone an exome or a genome because that has not been proven. So in other words, on the clinical trial side, getting comprehensive data can be extremely meaningful for the current decision, for the trial evaluation and monitoring, but also for subsequent line of therapies to create the evidence to ultimately drive clinical utility that then can be or should be covered with appropriate evidence in clinical practice. So it's very important to distinguish those two things because it's really different paradigms.
Okay.
I see Cheng Chui. Hey, Cheng. When data from multiple modalities are used to develop a biomarker, could they challenge this to validate because of requirement validation is that, yeah. Did I answer your question? Yeah. I just
Well,
wanted to ensure that it is one of the most challenging things because ultimately when you do an evaluation, you also have to trim off some data at some point. If there's no signal, okay, it's good to have the data. And everything in drug development is about timing and moving forward. And if you have to go back and add another modality, that might actually take longer than to assess it. I would say if we talk about real smart trial design, it might be that in the early phase, you might apply multimodal elements for, let's say, the first 10, 15, 20 patients. Then you see responders and non-responders. You look at that ad hoc, and then you use an adaptive trial design. And then in later phases of the study, you may not need to do all of it, but that fully depends on the trial and how that's progressing.
yeah. So Chris has a question here about a single cell DNA genomics platform that's being used in hematological cancers, but not solid tumors. And he asks, isn't it useful in solid cancers, or is the relevance of the information limited?
Yes and yes. So first of all, single cell RNA-seq or single cell sequencing technology in general means you dissociate the cells and you sequence them and you obtain data from every single cell. Number one, that's an extremely rich data set. And there's even some technologies that allow you to spatially resolve this so that you know where are my mutations distributed and what are the findings. So more data is not always good. It's also creating a few more problems, but it's in principle possible. Now with liquid or hematologic malignancies, since they're by default dissolved and mostly single cell, flow cytometry, flow sorting, and individual cells, it's just a little bit more amenable technically. But these technologies have been applied to solid tumors. And in fact, with Boston Gene, we have invested quite a bit of effort to sequence 25,000 cell lines and then create, based on these files, a synthetic tissue by combining multiple cell types together. We call this algorithm Cassandra. And this algorithm can then emulate single cell resolution from bulk RNA-seq data. So that solution overcomes the issue of the additional price point of single-cell RNA-seq, so we get near single-cell resolution. Now, we demonstrated in multiple publications the added value of that. You can do tumor microenvironment characterization. You can really distinguish what are the different expression patterns in certain tumors. Now, that's still considered, from a clinical perspective, emerging evidence. We have many trials ongoing, some with the Parker Institute, the Radiohead study, like multiple different studies to try to illustrate this. Why hasn't this caught on? I think it has not caught on in the broader sense that every single center is doing it because you need extreme compute power. You need to have a bioinformatics team that can manage and handle this type of data. To my knowledge, that's mostly restricted to certain research laboratories in tertiary academic medical centers or to the industry. So I would say it's a combination of technical challenges and compute or bioinformatics skills, why that hasn't been adopted in a little bit broader sense.
Would love to learn more about it at a later time and don't want to preoccupy that. But my question was actually related to Mission Bio's DNA, a single cell platform, and not the 10x RNA-seq
Got it.
that everybody talks about. Because the DNA is more or less what doesn't change in a way, or which is kind of the status of the different clones. But
it is definitely of interest in particular because the DNA level provides very, very important insights when you go at the single cell level between ploidy, meaning the copy number alterations that we just had, and then clonality. So does the tumor really harbor BRAF in every, BRAF mutation in every cell? So clonality and then diploidy, and both together form basically a purity assessment. So if possible to resolve this spatially and at the single cell level, that might actually be able to overcome this purity, ploidy, clonality issue. And then you can really assess this. I haven't seen a good clinical use case for it other than to make mutation calling more reliable.
right, my question was going into the direction because we talked about pancreatic cancer in the earlier phase, and that the tumor content may be limited, and with the single cell genomics approach, it should be possible to really distinguish these are the normal cells, or these are the normal, which likely are fibroblasts or others, and these are really the mutated tumor cells, and therefore having a very basically a very accurate assessment of the real mutations, and also being able to quantify actually is this just one clone, are there multiple clones, and what's the co-localization of those different mutations in those particular clones, to have an exact idea on what the sensitivity, or a better idea on what the sensitivity profile might look like.
Where do I sign? I absolutely, yes.
Yeah, let's talk offline. We also have working on something different at Sage Medics that you might find interesting. Also, es würde mich sehr freuen.
Wonderful, thank you.
Well, similar to along those lines, I guess, in the chat, and it uses the words
Yes.
along those lines, is it worth doing omics at multiple sites, given tumor heterogeneity?
Amazing question. I worked for years with Kamila Naxarova. She's a colleague here in Boston, and her entire career and life in a professional sense evolves around tumor phylogenetics and multi-regional sampling. And the amount and level of details of insights that you can get into tumor dynamic led to something that we coined internally clading. Certain clones metastasize to certain elements. She has published many papers on, for example, do metastases metastasize? Is a colon cancer seeding to a lymph node, and is then the lymph node seeding to organs, or is the primary tumor seeding directly to other organs? So when you do these phylogenetic maps, do multi-regional sampling within the primary tumor and within metastatic sites, and a recent paper, even peritoneal carcinomatosis, you get these trajectories that show that certain clones metastasize to certain regions, and these are called clades. That is completely unexamined, clinically absolutely ignored. And you know how everyone calls it typing, staging, grading? These are the three axioms, and now genotyping. We added one additional one, which would be clading, if we could just sample more. Now, the work has now expanded to melanoma, breast cancer, colorectal cancer, and most recently cholangiocarcinoma. Cholangiocarcinoma is a particularly heterogeneous tumor that has, multiple different clones within the liver, but then when it metastasizes, you can actually trace those elements into the periphery. One of the most clinically impactful elements to keep in mind is when resistance mutations emerge in these heterogeneous tumors, it's very important to know what can you detect in the peripheral blood, and is that fully reflecting the complexity of these tumors. So rebiopsy studies is definitely an approach to attack this problem, and these multi-regional elements or characterizations are deeply insightful from a biological perspective, but again, it remains to be determined when and how to use this.
Okay. So backing up, somebody asked if there's any updates in our regulatory environment and CDX regulation, if any.
Oh yeah, that's a personal favorite of mine, and I can find a brief statement that we put out as a white paper jointly with a large consortium with Friends of Cancer Research, and I share one of the sentiments in there. It's very, very important to me. So the big genetic companion diagnostic, let's say, treasures have been found. BRAF, EGFR, etc., you name it. The slices of the proverbial pie get smaller and smaller. NTRK, I don't know how many on this call have seen NTRK fusions. I witnessed a few in my decades of practice. It's an extremely rare event. Now we're talking orphan diseases, basically. So a lung cancer with an NTRK fusion or a pediatric tumor with an NTRK fusion, these are exceptionally rare. Now, what I wish the regulatory landscape would adopt is a certain relaxation of the requirements to prove that, let's call it analytical compatibility. So right now, there's a one-size-fits-all approach, but when it comes to orphan diseases, maybe you don't need 30 or 40 or 50 cases, but it would be sufficient to have 3 positive, 3 negative, if they have been confirmed by an orthogonal, like let's say approved diagnostic. So we put this together as sort of a white paper, and I put it in the chat to you so you can take a look. There are actually a few other considerations that should be done. So what I think might work and it will help access is to relax these very stringent requirements for otherwise already existing companion diagnostics. I think that would be my regulatory wish here.
Okay. Okay, there's a question. Could you comment on AZ Serena-6 trial? I can't pronounce that. You probably can.
Camizestrate, yeah.
Yeah, where surveillance for emergent ESR1 is being implemented, how practical or sustainable is this surveillance in patient care?
I know of the issue. I know that it's very controversial, but I do not feel confident in arguing for or against this. I think this is probably more a question for a breast oncologist. I know that the emergence of ESR1 mutations and how they're detected can be very, very tricky. I think if peripheral blood is used for this, we get to this point that I find really a challenge with circulating tumor DNA. That is, if the mutation is present, okay. If it's absent, we don't know if we just didn't detect it or if it was truly not there. I think that part, overcoming that, is currently a challenge in this context that I haven't seen anyone provide a sufficiently reliable solution. So that's all I want to say about this.
Okay, so I see Chris has another question. He says, who is working on phylogenetic cancer maps? Because we found very high correlation of primary and metastatic tumors for local spread, while correlation with hematogenic spread was still about 50%.
So the collaborator that I mentioned is Kamila Naxorova, and she works with a lot of different laboratories. So I know that at least 3, 4 other groups are working on it. But it's a I would call it even an emerging field. I don't know if that answers your question, but I think we will see a lot more over the next few years.
Okay, if you could just put the name into the chat, then I can look her up. So
I did. I think it's
Yeah.
yeah.
It's there. Yeah.
Okay, fantastic. Vielen Dank.
Thank you.
Okay. I don't see any other questions that anybody has. We've got a couple minutes left. And I see there's few links in the chat. And I don't see any, okay, let's see. Okay. So there's a question, hypothetical high-risk patient. If the patient would like to predict micromets and HSPC to CRPC development, what type of testing would you recommend?
Yeah, it's very tricky. This is something that is currently emerging. That is, I think the question is hinting at, some of the RNA expression profiling based assays. There are a few commercial solutions out there. I do not know the current state of the NCCN guidelines and whether they recommend it. So my quick take would be I would go to the NCCN guidelines and look at the expert panel, what they currently recommend, to just get sort of a more peer approach. If that doesn't lead to anything, I would need to go into the literature and double check the evidence.
Okay. All right, I do not see any other questions. Okay,
Thank you.
one
Thank you
last
very much for
question.
having me.
Ari Ackerstines, one last question. How do
yes.
patients typically find these tests, identify when it's relevant to get them? That's a good question. How do they find you?
So I think most of the patients that find us particularly is through some of the public webinars and elements here. I think we focus, however, I would say 95% of our work goes through oncologists. And I think most of the oncologists that send to us appreciate that we deliver a bit more than just the standard results. Meaning we typically don't test the first-line therapy patients. We do not test the initial non-small cell lung cancer that just presents. We test patients in third or fourth line. We test patients that have excruciatingly rare sarcomas or, fourth line or fifth line, no standard of care available, orphan diseases. So these exceptional slices that I mentioned earlier, that's typically where providers say, what else could we do? And then the word of mouth has it like, have you tried Boston Gene? And I know I see Michael and Rihanna on the call. We have helped. This is by no means we find something for everyone, but we find in certain settings certain elements that really change the course of therapy dramatically. And yes, this is a bit more expensive than normal. It is not always covered by insurance, but for certain settings, when applied meaningfully, we have seen some exceptional responses. So definitely a good question, but I would say very comprehensive testing is certainly not something for the broad population, but it needs to be applied in a meaningful way.
That's interesting that the oncologists are actually recommending this. I didn't know that was the case. I thought it was kind of up to the patient to go demand it. Can you talk a little bit about the cost? I guess insurance probably doesn't cover it, I understand.
Yeah, that's correct. I mean, it depends, we have a financial assistance program that is by household income. It's very competitive, similar to other vendors in the space. The part that I think is very important to keep in mind is the following. It's not just about making the finding. I showed you the little treatment timeline in my presentation. What I think is really important is that we put our findings in the context of care. Meaning, I'm sure you've seen, we found an NTRK fusion that means you can get a certain inhibitor. It's almost like a canned paragraph that is just dropped in certain reports. We would not do this. We would have the treatment timeline, and if the patient received that therapy, we would not recommend it again. It's still that finding, but in the context of care, it might actually lead to something entirely different. So this contextual interpretation is, I think, what some of the providers really appreciate, because it's very, very hard to keep up with these additional, elements of complexity or new findings, etc. So that is almost, I think, at least from the providers that I spoke to, they appreciate that. And if there's no main smoking gun, we have additional pages and additional pages that can help hopefully identify something actionable.
And I
Okay.
think as in the past, if anyone ever has any questions, Brad's always really good at referring patients or their providers to me, and I can help. But if you bucket us as a clinical test, we're all using the same billing codes as like Tempus, Caris, so forth. So you know, we do bill insurance, we do bill Medicare, but I can always take that offline if Brad has all my information. But it's just michael.hensley@bostongene.com if you have any questions for clinical.
Yeah, Michael, why don't you drop your contact into the chat? And I'm sure people may have some follow-up questions. I see we're at the top of the hour, and I want to be respectful of your time, Joe.
Mhm.
So unless you've got any closing comments,
Well,
think-
Helen asked a question or made a comment here. So the question of, you know, what other tests could be useful, I think that is something, you know, you must discuss it with your provider because they help with the immediate care. But just asking, is there something else that we could do, often opens up, you know, a conversation. I would recommend that. But thank you. And
Okay.
thanks a lot for having me.
Okay, thanks very much.